Okay, up to this point, we have been learning about mitosis and cancer and cell division that happens in the vast majority of our body cells. Now we're going to talk about a different kind of cell division called meiosis. This is cell division that happens two times. In mitosis, the cell just divides once. In meiosis, the cell division happens twice. And at the end, we get four sex cells. Sex cells are called gametes, and that would be things like sperm, egg, or pollen. And those gametes have half the original number of chromosomes. At the end of mitosis, we have two cells that were identical to the original cell. At the end of meiosis, we have four cells that have half the DNA. Because the only time meiosis happens is to produce sex cells, the only place that meiosis occurs is within the testes of the gentleman or the ovaries of the ladies. And by now I hope you remember that the only cells in the human body that have 23 chromosomes are the sperm and the egg cells. All other cells in the body have 46 chromosomes. If this were a different animal, if we were talking about a horse or a dog, the number of chromosomes in their sperm and egg would be half of whatever their normal chromosome number is. So if a horse normally has 78 chromosomes, then it would have half that amount in its sperm or its eggs. That way, of course, when the sperm and the egg come together, they are haploid organisms. They have half the DNA. They form a single cell called a zygote. That's a picture of a baby, basically, with the correct amount of DNA. So sperm and egg are haploid. The zygote is diploid. It has two sets of all the DNA. So in a human, the sperm and egg have 23 chromosomes, and then the zygote, this baby, has 46 chromosomes. The zygote then begins to divide through mitosis. So again, the only time you have meiosis is to form sperm or egg. Mitosis forms all the other cells of the body. Now, for those of you that did the meiosis puzzle in class, you saw that there were some very striking similarities between meiosis and mitosis. If our old phrase to remember mitosis was IPMAT, this time it's IPMAT-PMAT. Notice that interphase only happens one time, and then prophase happens twice, so we call it prophase 1 and prophase 2. Metaphase happens twice, and so forth. So IPMAT-PMAT. As many of you figured out, meiosis starts out the same as mitosis. We have an interphase stage. That's the germ cell that's going to become the sperm or the egg. It has 46 chromosomes. The DNA is in chromatin. Then during prophase 1, the nucleus disappears, the centrioles move to the side, the spindle fibers appear, and the DNA wraps up to form those bivalent chromosomes. Now, what's not indicated in this picture, unfortunately, is this sentence down below. The homologous chromosomes at this time find their partners. So for example, on the drawing, the two pink chromosomes would come together, the two green would come together, and the two orange would come together. The process of finding their partners is called synapsis, and that is a vocabulary word that I'll expect you to know. So synapsis, finding their partner, happens during prophase 1. They don't line up yet, they just find their partners. Now, in the last unit, we mentioned the concept of changing DNA through a process called crossing over, and we said this gives us new chromosomes. It's not bad. It's a way of increasing genetic variability. Crossing over happens during prophase 1, when the partner chromosomes find each other. It says here, during synapsis, neighboring chromatids, the little sections of a bivalent chromosome, may wrap around each other, break off, and then reattach to a different chromatid. This is called crossing over, and this leads to increased genetic recombination, so that there are chromosomes now. The two chromosomes at the bottom have never existed before in the world. Now, if interphase and prophase were the first two stages, then of course metaphase, IPMAT, comes next. The thing to notice about metaphase in meiosis is that instead of lining up one at a time, they line up as pairs. They line up with their matching partners, called homologous pairs. So during metaphase 1, the chromosomes line up in pairs, not in single file as they did in mitosis. Okay, during metaphase, the homologous pairs are lining up is partners down the middle of the cell. The part to understand is that they're lining up randomly, which means in the drawings that you're looking at, the pink and blue can be on either side. It just depends. So that increases genetic variability. It's got a special name, the random migration of chromosomes to different poles during anaphase 1, which is what is about to happen, is called independent assortment. Basically, that means that the eggs and sperm that are going to be formed because of this are very random. Sometimes blue chromosomes will go to the left side, sometimes blue chromosomes will go to the right side. And when you have 23 pairs of them, you get a lot of genetic variability from that. That's called independent assortment when they go to different poles randomly. As indicated, the next stage is called anaphase 1. Notice this time the part that makes anaphase different from anaphase in mitosis is that the entire chromosome is moving to the right or the left instead of just one side of a chromosome called a chromatid or a monovalent chromosome. This time the entire chromosome migrates either to the right side or the left side. So the homologous pairs are being split. Notice also that we still have 46 bivalent chromosomes, but that the cell is about to divide, the nucleus is about to divide. That's what we call telophase 1. And notice that once that cell divides, there's only 23 chromosomes in that left nucleus and 23 chromosomes in that right nucleus. They return temporarily to the chromatin form. And so now this is when the chromosome number cuts in half. Now the weird part is that cell division still has to happen again. The reason it has to happen again is that the chromosomes are still bivalent in that nucleus that has 23 chromosomes on the right or the left. They're still bivalent and we need to make them monovalent. So the cell is going to divide one more time to produce 4 cells that have monovalent chromosomes. Okay, so now we've gone through telophase and cytokinesis. Those 2 cells that you're looking at are still telophase. It says now there are 2 cells with each of the original number of chromosomes, I'm sorry, with half of the original chromosomes, 23, but the dividing continues. So I'm showing you a cell on the left and a cell on the right. Those are cells that have 23 chromosomes now. Now they continue to divide, so they go into prophase 2. Prophase 2 looks polka dotty, but notice they only have, in our example, they only have 3 chromosomes, whereas they used to have 6 chromosomes. Of course, the next stage would be metaphase 2. Metaphase middle, they line up in the middle. Again, just the half number of chromosomes. Of course, anaphase pulls the chromosomes apart, so now they were bivalent, and now we're back down to monovalent chromosomes. Now don't get thrown off here. It says, notice the temporary chromosome number here. We had bivalent chromosomes. We had 23 bivalent chromosomes. All we did was pull those chromosomes apart, so now we have 46 monovalent chromosomes. As soon as the cell divides, right here in telophase and cytokinesis, as soon as the cell divides, the number goes back down to 23 monovalent chromosomes. So look at anaphase 2 again. You doubled the number sort of just because you broke apart the bivalent chromosomes into monovalent chromosomes. So it seems like they have 46 chromosomes, but as soon as you split those 2 cells in half, then each of the 4 cells have 23 monovalent chromosomes. So now the part that you have to understand is that what those 4 cells are at the bottom. If this whole process happened in a female of any species, then those 4 cells are eggs. If it happened in a male, those 4 cells are sperm. We'll repeat that in just a second. Here's a quick diagram that skips several steps, but it shows you sort of generally what's happening in meiosis, and that you start with 2 chromosomes, and in the end you end up with 1 chromosome in each of the cells. What are those 4 cells at the end of meiosis? They're called gametes, and that's sperm or eggs or pollen if it's happening in a plant. Now, as a general FYI, I don't think I'll ever test you on this, but at the end of what they call spermatogenesis, which is what's happening in a male to produce sperm, there are 4 sperm. But at the end of oogenesis, which is what's happening in a female to form her eggs, only 1 of the cells actually becomes an egg, and the other 3 disintegrate. The way the cell division happens, the majority of the cytoplasm and the cell organelles end up in 1 egg, and that's the egg that goes on to maturity. So females really only get 1 egg out of the cycle, and the 3 others, which they refer to as polar bodies, disintegrate. At this point, we just need to mention why meiosis happens in organisms. Bacteria reproduce asexually. The prefix a means without, so bacteria reproduce without sex. They basically split in half and clone themselves, and that has been extremely effective for bacteria. The disadvantage to cloning yourself is that you all have identical DNA, and if one disease or one event wipes out one bacteria or one clone, all the other clones are also susceptible because they're identical to the one that got killed by whatever disease or whatever event. Asexual reproduction is a disadvantage because everyone is a clone. Sexual reproduction is what happens with meiosis and the formation of sperm and egg coming together. Sexual reproduction increases genetic variability through several methods. The independent assortment of homologous chromosomes, remember we said that that's how they line up together and then go randomly to opposite sides. We also get crossing over of DNA during synapsis of prophase 1, and that increases the variability of the chromosomes. And then there's also random fertilization. We never know which sperm and which egg are actually going to come together. So that increases genetic variability. Why is that such a great help? Genetic diversity is an advantage, and we get genetic diversity through sexual reproduction because the environment is always fluctuating. And in a fluctuating environment, maybe the environment might be changing because of a natural disaster or a species moving around to a new environment. There might be new food sources, old food sources might disappear, there might be new diseases, climates might change, we know that's happening right now, new predators come in, et cetera, et cetera. The environment is always changing. The more variability we have, the greater the odds that some of the offspring who are born will survive those new conditions that may have been unfavorable to previous generations. When you look at the puppies here, those 3 puppies, if they weren't cared for by human beings, if they had to actually live in the wild, their different coat colors give them advantages in different environments. If that white puppy were born in a tropical environment, it might be at a disadvantage. It might have a harder time hunting because it was easily spotted, whereas the other 2 would be better camouflaged. Vice versa, if they were born in a polar climate where the snow is present most of the year, obviously the white puppy would be at an advantage. So having that diversity increases the chances that some of the offspring will be able to survive.